everyone. My name is Kate Warner. And we're very pleased to be giving you the first guided walk through the new UniProt website, which is in its beta version. I'm one of the curators for UniProt, and I'll be presenting this webinar along with my colleague Aurelian, and I'll just let him introduce himself.
Hi everyone, yes, I'm Aurelian Luciani, and I'm one of the developers in the UniProt website team. And so I'm going to be covering a later part of this webinar, and yes, I'm going to let Kate start with that presentation.
So the way that we've structured this presentation is I'll start off by giving you a bit of background about UniProt and about how the website was redesigned. Then I'll talk about the changes that we've made to the homepage, to search, to our results pages and our entry pages. And then Aurelian will go through the improvements that we've made to our tools. And then we'll finish off by giving you some information on how you can give us feedback to help us improve the new website. So Aurelian and I are just two people in a very large consortium, which is spread across 3 institutes. There's EMBL-EBI in Cambridge in the UK, PIR in Washington and Delaware in the USA, and SIB in Geneva in Switzerland. So I think I can safely say that probably everybody in the consortium has had some sort of input into the redesign of the website, but it's really been the development team that has been working really, really hard to develop the new website with lots of user testing and lots of redesigns and formatting to get it ready for the beta release. So for those of you who are unfamiliar with UniProt, UniProt is a comprehensive, high quality and freely accessible resource of protein sequence and functional information. So we provide a lot of data about proteins, including sequences, isoforms, function, involvement in disease, structure, data analysis tools such as BLAST and Align, and that's just a few of the vast amount of data that we provide in our database. So what the UniProt website aims to do is to enable the scientific community to make full use of all of this data while being adaptable enough to meet the evolving needs of our users as new technologies emerge for data generation. So the core principles of the UniProt website in regards to the old and the new website is that the information that we provide should be easy to find. It should be customizable, so you should be able to view the data you want to view and to download which data you want to download. You should be able to easily explore our database, analyze the data that we have there using our tools. We provide different types of visualizations to help you interpret your data. And of course, we enable you to download in different formats so that you can carry out your own analysis. So this is a timeline of the evolution of the UniProt website. So you can see that the first version was released in 2008, and it's gone through quite a few phases of redevelopment. But in 2019, a new design was put in place for the next version of the UniProt website. And so since then, the developers have been working really, really hard. And now we're at this point where we released the beta version of the new website yesterday. And so it's ready to use in the hopes that in 2022, the new website will replace the old website. So why did we want to redesign our website? Well, so for the new website, what we've attempted to do is to build upon our core 6 principles that I showed you previously, and also make sure that we're fulfilling the website aims. So what we wanted to do is to give the website a really new look and feel, while improving the user experience and enhancing the ease at which people can explore our protein data in our databases. So to do this, we turned to our user community and implemented a user-centered design process by collecting information on the different ways people utilize our website. So this included examining web analytics, carrying out user workshops, surveys, questionnaires, focus groups, and interviews. And this was in order to document user requirements and ideas and measure their workflows and behaviors so we could identify opportunities for improvement. So participants in the user testing were from various fields, including bioinformatics, proteomics, genomics, molecular biology, and more. And we carried out extensive usability testing. So this redesign went through several rounds of user testing and redesign until we got to the beta version of the new website. So this can be viewed at this address. You can also access it through the old website. There's now a banner there that you can select. So this is what the old website, the current website looks like. You can see that the colors are quite muted. The text is quite small, and it's often very hard to see which sections of our database you're looking for. And so if we compare it with the new website, so this is what the new beta version of the website looks like. You can see that now the search bar is much more larger and prominent, as this is where most users start using UniProt, and this is the place that we advise most of our users to start. The databases, so here are our core databases. Our data sets are displayed in a way that makes them easy to notice and quick to access. So for example, in our core databases, they're shown as these colorful tiles with large text. And following user feedback, we've also implemented a need help section. So this section here, and this is where we've gathered all our help content. So in this section, we provide help and training content, display a panel with shortcuts to the help center, which is here, tutorials, webinars, training, and ways that you can contact us directly. So let's take a closer look at the search bar. It's almost similar to the one that we currently have on our website with many improvements. So you can still select the database that you want to search, and there's an advanced button, but now we have this list option. So this is where you can put a list of UniProt accessions, either from UniProt or from other resources, and then you can map them to the proteins in our database. So this is actually a shortcut to the ID mapping tool, which is also in the tools section. And we added it to the search bar following user feedback, which said that the users were expecting this option to be here. One of the differences is that now if you click advanced, the window loads from the side rather than from the top down, and you can select the database option so that you can perform an advanced search in your database of interest directly from the home page. So if you click this drop-down menu, you can select any of our databases and directly perform an advanced search. For the purposes of this demo, though, I'll be using the UniProt KB database, which is our core database. So let's go through an example and say that we're looking for the pink1 gene. So here I'm going to write pink1 in gene name, and let's say I want to filter my results so that it only returns entries that have an active site. So if I click the drop down, I can search for the field similar to the old website. Click here and click search. So this is the look of the new website results page. By default, the results are displayed in our new card view, and entries are ranked by the amount of content. So you will always see those with the most information at the top of the search. We've also added additional filters on the left so that you can filter down all of these results to find the entries that you're interested in. So we've added that you can search for proteins by having certain features, by its existence, annotation score, and sequence length. You can also filter by status, as you could on the old website. So you could only look for reviewed entries, entries that have been reviewed by a curator, or unreviewed entries. So these are entries, sequences that have come into UniProt but are waiting to be reviewed by the curator. So each of the entries forms this card known as an entry card. And what this is it contains a quick overview of all the information that is in the entry, as well as links to the different sections within the entry. So here we have a bit of information about the protein name and the accession number. I can see that it's a SwissProt entry because it has a gold page with a star. We provide the keywords, and here are the shortcuts to the different sections within the entry. So let's say I'm interested in disease. I can click here. It will take me to the entry page and then scroll straight down to the disease and drugs information. And I can see that this protein is involved in Parkinson's disease. For a more detailed view of the results and to enable you to customize the data that is displayed on the results page, you can actually switch to the table view at the top of the results page. So it's this button here. Although be advised that the developers have found through user testing that this is a problematic representation, and they are probably going to change it to something more obvious, probably full text names for both views. So let's switch to a table view. So in table view, you can see that some of the columns are sortable. So you can click on their headers in order to sort them by the corresponding values. You can also customize the data by clicking here, similar to the other website, but now the customization loads from the left, and you can see within the same window, and you can see the changes that you make directly in the window. So let's say that I'm interested in sequence length. I can put in length and select it, and you can see that it's already added it to my results table. But let's say I don't want it there, I want it on the left. To make it easier to look through, I can move the column. So it's really customizable. And when I close, you can see there's the length there. So at the top of both of the results pages, we still have a download function. Now, rather than it, but the download now contains many more options than on our old website. So when you click download, actually first, so let's say I want to select these entries. These are the entries that I want to download, some SwissProt and some Tremble entries. When I click download, it loads from the left again. I can either download the ones that I've selected or download all of the entries in my table. I've chosen a faster format, and if I want to see what that looks like, I can preview it, and now the preview opens within the same window, which is really convenient rather than opening in another page. In addition, if I choose the Excel format to download, you can see that all the data that I download, I can customize just like I could on the results table. So if I want to, let's say I don't care if it's been reviewed or not, but I also want to add an active site. I can add the position of the active site, and I can preview, and this is what it will look like. So there's the active site column, and there's the active sites. There's no active sites in these ones because these are Tremble entries, so they're waiting to be annotated. So let's explore an entry a bit more in more detail and see what that looks like on the new website. So I'm going to look at the pink one mouse entry. So at the top of the page, you can see that this is a SwissProt entry. So it has this gold page and a star. And what this means is that all the information in this entry has been assessed by an expert curator such as myself. You can see the accession number, the ID and name, protein name, organism, gene name, sequence length, its evidence, and its annotation score. So 5 out of 5 means it's been very well annotated. There's a lot of information within this entry. We still have the tools at the top. So we have blast, we have download. You can add the entry to your basket. You can add a publication, and you can give feedback if you find that any information is missing or needs updating. We also have tabs which allow you to switch between different views. So you can switch from the entry view to a feature viewer, which gives all the sequence features, lists all the publications and entry history, which you can download. We also have a new tab called External Links. This contains all of the cross-references to other databases that we have within our entry. The navigation bar is still on the left. It allows you to, except now all of the sections are listed right at the top of the page. So you can see every single section that has been annotated for this entry, and you can easily move between them. Also, as you scroll up and down, you can see that the navigation bar stays in place. So if you go here and you want to go straight back up to function, you can click here and it will take you straight up to the top. Evidence tags. So these are our evidence tags. You can see that they're tags that have been added by a curator because they've got that SwissProt page. So these have been reviewed. These tags have been reviewed and added by a curator. And so they contain a lot more information. The experimental evidence tags, that's what I mean. So for example, in this function comment, I'll start again. So all of the data that we have in an entry, we try to provide evidence tags to show users where that information has come from so that they can follow it to make the assessment themselves if they wish to. So in this case, in this function comment, we have information, and we've provided the PubMed IDs where that information has come from. If you click the evidence tag at the end, you can see now it comes up with all of the information about the publication with links to PubMed and UniPMC, but now that you can view the abstract directly within the entry without having to go to these websites. And you can also see what other entries the publication has been mapped to. The similarity tags. So these are information that's been propagated from another SwissProt entry. Will still give you a link which takes you to that organism and that entry. So in this case, it's a human entry. And just for the purposes of showing you what the different entries look like, this is a Tremble entry. So I'll show you what a Tremble entry looks like. It has this grey page, which shows that it's not. So this is a sequence that's come into our database, but it hasn't been curated by one of the curators. So all the information has been added through automatic annotation. You can see by this evidence tag also, you can tell it's automatic annotation because it has this grayed out page. And when you click here, it will tell you which rule this relates to. So if I go back to the SwissProt entry, another nice feature that we've added is we've actually embedded our PropVista feature viewer directly into the entry so that you can both see the position of the feature in the sequence and read about the feature at the same time. So for example, if I move down to function, So here is our SIGPROP Vista sequence viewer. You can, if it's a long list, you can select which type of feature you're interested in. So something such as active site, and it will bring up just active site. Or alternatively, if you just want to look on the sequence, you can click. So if I click here, so it tells me what that is. It's this active site. Alternatively, you can actually click on the table, and it will show where that position is in the sequence. And finally, another thing that we've added, I'll just go to a different. So just to show you, we've also embedded our structural information in our entries. So you can see them directly within the entry rather than having to go to the feature viewer page. You can select the source, PDB or AlphaFold, and the method. And just like on the feature table, you can click between, you can zoom in and out, and you can click the different things to look at what these different structural information is. And we also have predicted structures from AlphaFold. This has very recently been made accessible through the UniProt website. So when they're available, they've been added to the entries. So I'm now going to pass over to Aurelian, who is going to talk you through the tools improvements that we've made.
Yes, thank you, Kate. So yes, I believe you can see the screen, the homepage. Yes. So now I'm going to talk about the tools that we have integrated in the UniProt website. So we already had tools before in the previous version of the website, and we still have them. So if you really like the blast or the align features that we had in the current website, we still have them and we improved them. You can find them through different places. You will always have access to them through the header in this section of the website. On the home page, they're also listed here. So we have 4 tools: blast, align. We also have ID mapping, or also known as search by list, that Kate mentioned earlier, and peptide search, if you want to search for specific peptides. And also we sprinkled those tools all over the website. So if you can, you will see that wherever you browse our UniProt data, you will have access to the tools. So here I have access to those 3 tools: blast, align, and ID mapping. At the moment, I don't have any entries selected. That's why they're disabled. But when I start selecting them, I can start sending them to the tools. So here I would blast this specific entry. And if I select a few of them, I could align them or map their IDs. Also, from within the entry page, as I mentioned, you can blast or align directly from here. Those work for any entry that contain sequence information. So that would be for UniProt KB entry, but also UniRef and UniPark entries, you would be able to blast them. So I'm not going to extend really about what UniRef and UniPark are, but you have more information here, and you can find previous trainings that our team has done about UniRef or protein clusters in the UniProt website, or the sequence archive, which is also known as UniPark, as part of our own data. So let's start with an example that was mentioned just earlier. So if I look for parking, I would get a list of UniProt KB results that are related to parking somehow. So I'm just going to take this first one. This is from human, and it's fairly well annotated. I can see here that it is manually reviewed UniProt KB entry. And so yeah, if I go and blast, I would jump to this form page, already pre-filled with the sequence in FASTA format. Also, I could just have gone, so I'm just going to copy this accession, but let's say that I can directly have come from this link and look for the accession here. And went so quick that it loaded that automatically, but I could search for any accession here, P05067, for example, and this would load the sequence here so that I can blast it. Just going to check. Yes, that's the one I wanted to blast. Okay. What options do I have? Here I could actually blast more sequences if I wanted to. At the moment, we can only blast one sequence in the current website, but in the new website, you can blast multiple sequences at the same time with this with the same options. The only thing that is going to generate multiple blast jobs that are going to run in parallel. The main choices that you have are quite common to what you have in the current website. So the main one would be the target database that against which you want to run the blast. So by default, we have UniProt KB reference proteome plus WISProt, which gives a fairly good trade-off of the number of results and interesting results and speed, because the bigger the target database will be, the longer the blast might take. So, for example, you could run blast against UniPark, the whole of UniPark, which is way bigger in number of sequences than UniProt KB and reference proteomes plus WISProt, but it might take way longer to run. So that is a kind of trade-off that you have to decide depending on the analysis you want to do. Also, one way, so this is something new. We can now also restrict by taxonomy. So if you know that you're going to be only interested in things, in sequences from primates, you could look for primates, and then you would be able to do this blast search only against primates, or you can add other taxonomies. That is a way to reduce the result set. And as before, we also have a lot of advanced parameters that might not be interesting for your own use case, but they are there and you can customize them if you need it. I can run blast. What you would get before would have been a page that would be loading and refreshing from time to time and eventually gives you the results. Now, what you get instead is a dashboard of all the jobs that you have run on the website before. And you can see the status of the job. So you see this one that I've just created is still running. It has been assigned an automatic name. But if I want to remember that one, I could say, okay, that is parking against UniProt KB. Well, this is whatever name I want, and then that way I can find it later when I come back to this page. That could be the day after. That could be in 10 minutes. The name is going to stay here, and you are going to be able to find your job easily. The good thing is now that you can actually continue browsing around, submit new jobs, explore the data, and here I have access to that dashboard also. With this toolbox icon, and I can see that my jobs are running. And if I continue browsing, I will eventually get a notification that my blast has finished running. Oh, well, it just opened now. But if I was browsing the website without looking at the dashboard, you can see that I get this notification here. Yeah, so let's jump to this the results. Okay, one thing I wanted to mention before is that also the jobs, as before, are limited to 7 days. So when you run a job, be it blast or line, we will keep it on the servers for 7 days, and you will be able to view the result for 7 days. After that, we will keep it in the dashboard for 7 additional days, but you will not be able to view the results. What it allows you to do is to resubmit the job at a later point by using this icon. So if I do this icon, I will be taken to the blast form with the same parameters I've chosen before, and I will be able to rerun the blast. An interesting thing is that if you have an interesting set of parameters or of an interesting sequence that you want to rerun maybe in 2 months or in the next release of the UniProt KB data, you might want to star it. If you star it, we will keep it and it will not be removed after 14 days. And so you can be able to come back to this page 2 months later and you will be able to rerun this same job to have more up-to-date data. Now I'm seeing the BLAST results page. This has been visually changed, but you still have access to the same underlying data. So I'm directly going to jump to this. Here, with the tools that we provide in UniProt KB, we're mixing tools that are quite known in the community, such as BLAST and Align, and we're running them in the background, and we're associating that with the data that we own at UniProt, and we want to provide to the people using the website. So we're mashing together the result of these tools and the data that we have here. But if you want to access the underlying results of the BLAST, if you were to run BLAST yourself as a command line tool, this is what you would get. So you still have access to the underlying data, and you can take it, you can download it here, and be able to run your own follow-up analysis. You have the information about the input parameters that this job has been submitted with, so that's the sequence I've submitted earlier and all the parameters. And also, if you want, you can also copy the API request and do that yourself on the command line, to rerun that at a later point. You have the overview of the results here. I have 250 results because that's what I've asked for. I've asked for 250 hits on the BLAST form. And so Kate showed you earlier the filters on the side of the UniProt KB results. You also have access to them here. So if in these 250 results, I'm only interested in the reviewed ones, I can have a look and filter only the reviewed ones. So yeah, that might be interesting. So I'm going to keep them for later. So I'm interested in this one, and this one, which are all the reviewed ones that are not isoforms, and I'm going to add them to my basket. The basket is also something that we had earlier in the previous website, and is still working in the new website. You have access to all the sequences that you've kept in your basket at any given time, so you can come back to them later and use them for further analysis. Okay, so what you can see here are histograms of the value specific to the BLAST search that you've run. So here you have the different scores of, well, you have identity, you have score and e-values that are actually listed here also. And if you're only interested in really high identities and high score, you can filter that and your result set will be reduced. So you see now I only have 51 results that are within this criteria. Okay, and I can see this also here. This is just a bigger view to have a bigger view of the different BLAST values that you get from your BLAST job. You also have information about the taxonomy in the result set. So you have a taxonomy tree of all the sequences in the result set. So here you can see that you have some mammalia here, primates, and if I close that, you have some other organisms. And if I filter on the side, you can see that we regenerate the tree only containing the sequences that you have filtered in this case. So let's say now that, just going to check that I covered everything I wanted to cover. Oh, yes. And now, if I'm interested, let's say in one of them, this one, and see how it matches the query that I've submitted before. So I can click on this visualization, which is a mini visualization, as Kate showed you before, the Prot Vista visualization that we have across all the different pages. And I can click on it, and it will show me the pairwise alignment between my query and the matched protein. So here, this is an overview of the alignment, and I can scroll across it and see which parts are conserved, which parts are different. And I also have access to the different annotations. So if this might not be a good example, I'm going to take a reviewed one. Let's say this one. And then I can say, okay, show me the annotations that are on this of a protein. I want the different regions. So, or domain here. I have this domain here, which is annotated, and I have maybe a zinc finger that we know has been annotated in this part of the sequence, and then I can look if I'm. Then knowing this annotation, I can possibly deduce if my query sequence might have the similar possible characteristics. Let's say now I'm looking at unreviewed, and I'm going to take one that really bad annotation score, a really low annotation score, not bad, sorry. And let's say I have this one, which is marked as uncharacterized. Obviously, if it's been written in the blast search, it probably means that it's quite similar to the one I've used to do the query. So I'm gonna add this to my basket also, and I'm gonna do something with it later. And I'm gonna also look for someone that would be, some sequence that would be human and unreviewed. So using this filter, you can see that I filtered by unreviewed and by human, and I have those two matches in there. So I'm also going to add them in the basket. So now in the basket, I have 6 sequences. I know 3 of them are reviewed, manually curated. 2 of them are, no, sorry, and 3 others are unreviewed. So I might want to select them all and align them, just to be able to compare them. So you can see now that I have used the other tool, which is align. I've used the output of the blast result in order to go to the other tool that we provide, which is the align tool. And it automatically filled the form with all the sequences that I've selected. I can also change the name of my job here. So my alignment, and run the alignment. Alignment usually is quite quick to run. See that it took a few seconds to run. And now if I open that, I can see that instead of having the pairwise alignments that I was seeing before in the blast results, now I have the alignments between the 6 sequences at the time. It's quite interesting to be able to explore the possible characteristics of the sequences by comparing them between each other, especially now that I have 3 of them that are manually curated and fairly well annotated. I can compare them. So here you can see I have the 3 spot one or reviewed one and 3 Trumbo ones or unreviewed. And I have this view, which is the overview, which is similar to what you had before, or we can see the whole alignment of sequences and browse, scroll through the whole alignment, or you can have the wrap view, which is something fairly similar to what you would get in the raw output of the align tool, and allows you to see in one page, but by scrolling, all of the alignments, all of the alignment data. What you can do is changing this visualization. By default, we're coloring the sequences by similarity, but if you're used to other tools, you know that you have other ways to color that. So that could be this cluster color scheme, or by physical property like hydrophobicity. And yeah, if you used to other tools, you will be familiar with the options that we provide to you here. Let's go back to similarity. Let's say now that I'm interested in modified residues. So I can look for that. I see that I have annotations for modified residues in those 3 sequences. All SP as SwissProt, which means they are reviewed, manually reviewed. And I'm going to show the ones for human. So here I have modified, let's switch to the overview. I have modified residues here, here, and there. And you can see in the other sequences, so that is the sequence I've decided to show here. But in those 2 other sequences that are also reviewed, I also have the same annotation. But in the 3 other unreviewed proteins, I don't have this annotation. So what does it tell me is that if I look at the conservation of this specific section of the sequence, I might think that this is a possible site where we might have a modified residue at this position. The thing is, it is not annotated, either because it is not the case, or it needs to be reviewed, or because it's not annotated yet. So if someone wants to do some studies and provide feedback and give us information about this specific sequence and this specific annotation, then they can provide feedback to us, and we can eventually pass that to curators, and they can integrate this data. So what's here, so just gonna, yes, so the parking sequence, it needs to be phosphorylated by pink one, the other sequence that Kate was talking about before, in order to function. And if it doesn't, if it's not phosphorylated, it might link to Parkinson. So that is something that might be interesting to people studying this. Let's see. I think I covered everything I wanted to show on the line. Let's quickly go through the other tools. I have also peptide search and ID mapping. So I've run some jobs before. I actually have access to them here. So ID mapping. I might want to, let's say this one. Okay, so if I have a list of IDs that I want to map to another resource, either within UniProt or outside of UniProt, that could be, I don't know, Cambo or whatever. Let's have a look at the options. That could be any of those resources. Then I could paste the list of accessions here, and this tool would be able to do the mapping. That could be from UniProt to other resources, from other resources to UniProt, or from UniProt to UniProt, depending on what you're interested in. So here in this example, I had UniProt KB accessions, and I want to map them to UniPark accessions, UniPark IDs. And this is what the tools allows me to do. And I also have peptide search. So it allows me to do an exact amino acid search against the UniProt KB database. So that can be quite specific. If you have a really specific section of the sequence that you want to find across our data, you can use this tool instead of BLAST, because it's going to be probably quicker and more accurate than doing a BLAST job. Yes, with that, I'm going to hand back the presentation to Kate so that she can close it. Sorry.
Thank you. Thank you, Irene. So I'll just go back to sharing my screen. So thanks for joining us. I hope you've liked the improvements that we've made to the website, and I hope this encourages you to help us to develop the website further, to cater it for our users. So our future plans for the website is to continue the user testing, to collect feedback for the beta release, and to release the new website in 2022. So we've only released the beta version of the site so far, so it's ready to use, but it's not perfect yet. So you can help us through providing us with really valuable feedback. So on the new website, you'll see that there's a little feedback section, which you can select and provide feedback within the same window. And if you would like to formally sign up for testing the new website and participate in future user research, you can register your interest here. And I think Aurelian has pasted this link in the Q&A box so that you can quickly go there and register your interest. So for further help and documentation, you can contact us, which is at the top of the page on the new website, this little message icon, or you can go to the Help Centre, which looks like this. And at the Help Centre, you can find links to training, guides, documentation. Of course, things like upcoming and previous training sessions and seminars are available on the homepage that I mentioned in the new Need Help section. So this section, which is on the web page, the homepage. Sorry. So a little bit of details about our funding. So we'd like to thank the people who have funded the redesign of the website and all of the work that we do at UniProt. And here's a bit of information about upcoming webinars. For UniProt, we have one coming up on the 17th of November, which is about programmatic access to UniProt. And this will be run by Aurelian and another curator, Emily. And this one isn't live yet on our training pages, but you can, if you're interested in finding out how to use and make the most out of the new website, I'd recommend that you sign up to follow a student's guide to UniProt, which is really helpful for anyone who's new to using UniProt. So that's all from us, and we'd like to thank you for joining us, and please fill out the survey that launches after the webinar, and we'll be answering any of your questions that are in the Q&A. So thank you very much.
Okay.
Okay.
I think we can start taking some of the questions. So I think the first two are related to reviewed, unreviewed, and how to see them and how to filter through them. So I think across the presentation we've been through this in multiple parts, but I can go over it again. Maybe I will start sharing screen again.
Okay. Yeah. Stop.
Yes. And so in the results, you can see a reviewed entry because it has the same color code as before, which is the golden symbol here or here. And as Kate mentioned, also unreviewed would have the gray page in this case here and here. And so if you want to filter by that, you can filter either here on the side, or in the advanced search, you have a reviewed, I think it's called reviewed, yes, reviewed filter here. So you can choose that if you do an advanced search, that would be a bit more complex query, you can include this in your query. So here I would want either reviewed or unreviewed, and that would, well, give this in theory the same result set. Yes, results unreviewed here.
Yes. Another one. Yeah, so you've answered that one, haven't you? Or once search reviewed.
Yeah.
Could you review how to view features tabulated in ProtVis in a 3D viewer? So I think this means the 3D structures. Yeah.
Do you want to take over and explain that one?
Okay. Yeah. So, I've still got the structure. Oh, I'll share my screen. That would work better. So this is our 3D viewer that we have currently. So this is in the structure section of the website. Just zoom out a bit. This is in the structure section of the website. as you can see here. So what we do is we link all the structures when they're available from PDB to the structure section and show those structures. So if you wanted to just look at PDB information, you could select PDB and you can actually go through the different structures and see where these positions are. So we have positions and we have links to PDB and PDB sum. You can even select the method. So if you wanted to only look at sort of NMR or X-ray, you can select between them and it will filter those results.
Maybe
also,
regarding the features, maybe you can switch to the feature viewer page. With
yes. So this is also available on the feature viewer, as Aurelian pointed out. So this might take a second. So this is so you can view all of the sequence features in this view rather than reviewing them within the entry. And you can see the structure is here. It's easy to see on this page, and it's just exactly the same. You can go between PDB or AlphaFold when a prediction is available and the different methods as well. And it will move through the different structures.
And one interesting thing that you can do here is that if you go up and select a domain, for example, or any kind of feature that is annotated.
Yeah. So if you would,
yeah.
So maybe if you wanted to select some sort of PTM or modified residue or something, if you select here on the feature viewer, it gives you information about that evidence. Oh, it hasn't worked with this one. It might be that you need to go to domains yet. So maybe if I select the sync finger, it will show you, it will align it with the sequence. And as Aurelian showed, you can zoom in, see the amino acids, see where those features align. And it hasn't shown up in this one, probably because it's in a different position.
Yeah, it's out of the mapping range, so it was not.
So sometimes if you click on certain positions, it will actually show up in the feature table. I think that's right, isn't it? Really. Yeah. So that's a really nice way to look at the information that we have in regards to the sequence in just one place.
Yeah, okay. Obviously, there's a mapping between structure and sequence, and sometimes the sequence is larger than the structure or the other way around.
Yeah,
and
because they're all mini little bits. It will show you the positions. So these are the positions within the sequence for the structures. But the PDB, the predicted structure, so the AlphaFold, that's for the entire. So if you clicked the predicted, that's why it looks. It zoomed in rather than out. So that's why it looks so big in the other. So this is the entire sequence that's been predicted by AlphaFold. So yeah, it shows you. Yeah. Okay. Let me go back to the.
So there's another question. UniProt stops at release 2021-03 for a while. What happened? So. Do you want to talk about the release cycle of UniProt, Kate?
Sorry, I've.
Oh yeah, sorry.
I've lost the question. Sorry.
So the last release is 2021-03, and it's the third release of the year 2021, and there's an upcoming one which should be 2021-04 coming up at some point.
So we've switched. I don't know if you've seen, but we've actually switched to 8 weeks now rather than 4. So there's a lot less releases within the year than it was. And there was some sort of production issue with lots of sequences coming in or something, which delayed our release. I can't remember what that was about. Do you remember, Aurelian?
Is that from a few years back when there's been the effort of deduplicating the sequences? I think it was before I was in the team, so I don't remember exactly what it was.
So UniProt stops at release 2021 for a while. So yeah, that was this year. There was a few releases that we skipped. We don't have a release in August. we do less releases because we're every 8 weeks now. And then there was some issue. I can find out what that is if you're interested, and I'll get back to you. But there was some sort of we had I think we had a large number of sequences come in through Tremble or something, and we had to.
You think that would be detailed in the release notes of the next release?
Yeah, I think if you look at the release notes, yeah, that's a good idea. I can't remember the exact reason. I remember it was explained to me, and I can't remember by our the guys who deal with importing sequences. So it should be on the release in the news section on the website, saying why we skipped a few releases. Yeah.
There's one
the next question, can we do molecular docking? I'm not sure about that. That is something that I would have to look at and get back to. If you want to email me directly, the anonymous attendee, about the molecular docking, I can get back to you. If you email me kwarner@ebi.ac.uk, and I'll put you in touch with the right person who would know.
Yes, so I think we covered all the questions so far.
Yes, I think. Yes. Can you please post the link for the 2nd of December webinar, please? This is in the chat section. Someone's posted some questions in the chat. Oh, Anna's already responded. Yeah, the webinar is not live yet, but it will be. Why is there not a separate database with the name such as UniProt, Swiss Pro? We sort of consider Tremble and SwissProt to be separate databases. UniProt, Swiss Pro, that's quite good. I don't know. Yeah, maybe you can make it.
They'll mix together because it's similar data. The annotation level might be different, and the annotation source is different because that's the separation between the two. But they can be considered as one depending on the analysis you want to do. Especially if you want to compare them, it's quite good to have them together, and that way you can compare a unreviewed or Tremble to a reviewed or SwissProt, and you can see them in parallel. So I think that's why they're coexisting together.
Yes,
but you can still split them whenever you want.
So we also have, the automatic annotation that we provide, that has been, some of the rules have actually been created by expert curators. So they are pretty good at predicting things like function catalytic activity and that sort of thing. So the automatic annotation rules can be very, very accurate, even though we haven't had a chance to look at the entries because we get so many sequences. We get hundreds of thousands of sequences being imported with every release, which we have to look at. So it's impossible for curators to look through every single one. So we put them all together, like Aurelian says. We add annotation using automatic annotation protocols, and we put them together so in that way you can use our analysis tools to compare them with SwissProt entries, and in that way you can probably work out functions and of course, if you have an entry that's a Tremble entry and you want it annotated, you can always contact us and we will do our best if their information is there to annotate the sequence if it's a Tremble one. Yeah,
actually, there's a question that came up just now about how can I be in touch? How can I get in touch with us? So there are multiple ways that you can find that. So that could be either on the website, that could be the beta website or the current website. You can look for the contact us section either in the footer or the envelope in the header. And reach out to us. Also, if it's about specific entry or if you have specific annotation information, you can go to the entry page. That same thing, either in the current website or in the beta website, and you will get a section at the top of the page where you can provide entry feedback. So if you want to provide the annotations as Kate mentioned just now, you can go on the specific entry and provide feedback from the entry.
Yes, I forgot that. You can add publication and an annotation in our new tool at the top of each entry. So if you want to add your publication because you found it's not attached to the entry, you can add it there and add any information that you want. So you could do that with a Tremble entry if you wanted to, or a SwissProt entry. Yeah. And thank you, Deepa Pandit, who said it's one of their favorite websites. That's really nice. So again, yeah, I think.
Yeah, if you have any feedback or suggestions about the new development, because it's still development in progress, so please reach out to us. You have on the new website, you have the feedback button on the right that you can use to give us opinion or tell us if something doesn't work as expected. We will reach out to you and tell you either why something is missing, or if we're planning on implementing it, or how we can fix it. And if we want to see more features, do reach out to us also in the same ways.